And you're watching Steven Loves Science.
Hello everyone, I'm Steven and you're watching Steven Loves Science. So I've already done a few videos on activities that occur inside of the cell, but we've never really had a chance to discuss the actual anatomy of the cell and the structures that compose it. So today we will discuss cellular anatomy and we will go over the various functions of different cell parts. Now a good place to start is the plasma or cellular membrane. And the cellular membrane is a good place to start because it is what separates the cell from the outside world. And what it basically does is decide what goes in and out of the cell. And this capability is known as selective permeability. Now not to get out on too much of a tangent, but the cell membrane is actually what is called a phospholipid bilayer. Now the key word is bilayer. And you cannot actually see this with a light microscope, but if you were to put on your molecular goggles or use an electron microscope, we can actually see that the cell membrane is composed of two rows of phospholipids. So let's demonstrate this on the board. Putting on our molecular goggles and zooming in at a very high resolution, what we would see are these phospholipid heads, and we would see these phospholipid tails. So right now what I've drawn are these phospholipid heads, and then right now I'm drawing phospholipid tails that are hydrophobic and they orient themselves away from the aqueous environment around them so that they are facing inward and the hydrophilic heads are outward. Okay, and I found this important because going back to what I said earlier about selective permeability, the way cells achieve this is by having proteins that may or may not be embedded into this bilayer. And these proteins regulate the passage of molecules to ensure that the cell is always in a state of homeostasis. So let me label that cellular membrane. So what's an example of one of these proteins that regulate the passage of molecules? So let's draw here in blue just a protein here. So this would be what is called an integral protein because it is embedded directly into the phospholipid bilayer. Okay, so let's say this protein has a job, and its job is to regulate the intake of glucose. So here we have glucose on the outside. Okay, and we want to bring glucose from where it is in an area of low concentration to an area of high concentration. So we want to go against the diffusion gradient and set by that. So this protein would bring in glucose and control the amount of glucose that's in the cell to ensure that the cell is always in a homeostatic state. Okay, so all cells have a cellular membrane. So just to get that covered, if a cell does not have a cell membrane, it is not a cell. Now, moving on, if we think about maybe the most important thing that defines a cell, you probably would think DNA, and you would be right. So all cells have DNA inside of them. So we're going to draw our DNA inside the cell. You know, this would be in its chromatin form. So I won't go into the details of how DNA defines what an organism is because I've done that in the DNA videos, but all cells have DNA, So right here, this is the DNA, and as I've said, in its chromatin form, and there are also some little proteins. You may have heard that DNA is wrapped around histones, so as I've drawn that in its chromatin coiled form. So right here, what this represents right now is pretty much any cell, and any animal, plant, whatever kingdom could look like this, because I haven't drawn a lot of the details. I've just drawn the DNA and the cellular membrane. Now, here's kind of the first major division in the living world, or at least from our point of view, and that is that some cells have a membrane around the DNA, and some cells don't. So, if I were to draw a membrane around here, you know, the DNA and the chromatin and the genetic material is now separated from everything else in the cell. So now when we have the membrane around the DNA, it is called the nucleus. So let's label this nucleus. So as I said, this is a major part of dividing, of organizing cells, because when some people looked at the cell, they saw a nucleus, and when some people looked at other cells, they did not see a nucleus, and they said, hey, this is a good way to classify organisms. So they called things that had a nucleus eukaryotes. I'm sorry, if I said, so they called these things eukaryotes. So a eukaryote is a cell that has a nucleus. In other words, is a cell that has a membrane around the DNA, okay? So right now we're dealing with a eukaryotic cell. Okay, so now if a cell did not have a nucleus, you were dealing with a prokaryote. And examples of prokaryotes, the two big groups of them are bacteria and archaea. Now archaea are really interesting, but we're not going to focus on them. We're going to focus on eukaryotes because they tend to be a little bit more complex and they also tend to be larger. But now let's go back to look at the anatomy of the eukaryotic cell. So we have our DNA, and we know that it gets transcribed into mRNA. So transcription happens in the nucleus, and you can see my video on transcription if you're not sure what that is. And that mRNA that is transcribed from DNA in the nucleus will then leave the nucleus and gets translated into proteins at the ribosomes. So ribosomes are these little complexes that can be floating all over the cell. So let's draw some ribosomes that are, just floating around the cell. I'm not going to get into the structure of what an actual ribosome is, or it's a highly complex. There's a very interesting Berkeley lecture on how the structure of the ribosome was actually discovered, and I'll post that in the description. So here we have a ribosome that's just floating in the cell. Okay, and this ribosome is floating in the cell, but as we will see in a second, they can also be attached to these other membrane structures. So this is a ribosome, and if all this talk of DNA transcription into mRNA and mRNA leaving the nucleus and traveling to the ribosome to be translated into proteins makes no sense to you, there are several videos where I go into that in detail. But what I want to do now is just focus on the different parts of the cell to kind of give a big picture of things. The ribosomes are where mRNA gets translated into proteins. Okay? So you can kind of view them as a place where information actually turns, so you can kind of view the ribosomes as the place where information actually turns into proteins, which can then be used anywhere in the cell. So anyway, we're at the ribosome, and as we've established, it's where proteins get produced. I feel like that's a major point. You can take that away. But if the ribosomes are just floating around, if they're free-floating ribosomes, then those proteins, once they're produced at the ribosome, will just float around in the cell, here in the fluid inside the cell, or what we call the cytosol. So you may have heard the word cytoplasm. It is coming from cytosol. So this is what a biologist would refer to when talking about the fluid of the cell. Is called the cytosol. So we know that proteins can be made in these free-floating ribosomes and just float around in the cell. But what if we wanted to produce proteins that are supposed to end up maybe embedded in the cell membrane, or maybe outside the cell itself? For example, the job of your pancreatic cells is to produce insulin to be used by the body. And those cells, if you're interested, are called the islets of Langerhans, and it's their entire job to produce insulin, which is a protein to be used by the body. So as we've established, cells produce things that are used by other cells and that are used by the rest of the body. So to do that, so how do we export proteins out of the cells? Well, to do that, we have to go to this membrane structure. So I'm going to go ahead and I'm going to draw that in red. Now I'm going to tell you what it is in a second, but I'm just going to draw this first. Okay, so as I've said, to do this, we have to go to this membrane structure that I've drawn in red. You can kind of view it as a bunch of tunnels, because it really is a bunch of tunnels, and I didn't draw it that well. If you want, you can look it up. I'll tell you the name in a second. So, right now we have this thing that is called the endoplasmic reticulum. And these tunnel-like structures eventually lead to something called the Golgi bodies, or Golgi apparatus. So I have the endoplasmic reticulum in red, and I'll do the Golgi bodies in blue. So these tunnels eventually lead to these Golgi bodies, named after Mr. Golgi himself. Okay? So pretty poorly drawn, but it basically looks just like that, and I'll tell you in a second what's going on in there. So this is all just kind of a big stack, or you can view it as a bunch of folded membranes together. And what happens is over here, some ribosomes are actually attached to this, or actually attached to the ER, or to the endoplasmic reticulum. So I'm going to draw some black dots along here. And these black dots are the ribosomes that are attached to the ER. So now we know that some ribosomes are free floating in the cytosol and some are attached to the ER. And the area of the endoplasmic reticulum that has ribosomes attached to it is called the rough endoplasmic reticulum. So you can call it the rough endoplasmic reticulum if you like to sound fancy, or you like to use big words, or you can just call it the rough ER. And there is also a smooth ER, which does not have ribosomes attached to it, but we'll get to that in a moment. Sorry, smooth ER. So you can kind of think of the distinction: a rough ER is going to feel bumpy and rough because it has ribosomes on the outside, but the smooth ER has nothing on it, so it will appear to be smooth or feel smooth. So these ribosomes are attached right here, and this is the rough endoplasmic reticulum as we established. So what does this have to do with the exporting of proteins? Well, I gave you a hint earlier. In our free ribosomes, mRNA gets translated into proteins, and the proteins just float around in the cytosol. But if we want the proteins to end up somewhere else, perhaps embedded in the membranes or outside the cell, this is where the endoplasmic reticulum and Golgi bodies come into play. Because now what we can do is, we have mRNA leaving the nucleus. Actually, let's go over to my diagram over here. Completely forgot about that. Okay, so we're looking at a larger image of what we had over there, so we can better understand the process of how proteins are packaged and exported in the cell. Okay, so what we have here, we have the nucleus, and the nucleus is going to give out mRNA. And that mRNA can then be translated by ribosomes that are on the rough ER. So what happens here, the mRNA comes to the rough ER and is getting translated on the outside of the rough ER. But as that protein is getting produced, it is getting pushed to the inside of the endoplasmic reticulum. So let's say we have our mRNA strand here is being translated by a ribosome, and what we're ending up with is our chain of amino acids on the inside of the endoplasmic reticulum. And when I said the inside of the endoplasmic reticulum, I'm talking about this area here. We're looking at a cross section. And those proteins will get pushed into the rough ER, and those proteins are specifically ones that are meant to be used on the outside of the cell or in the cell membrane. So those proteins end up here. And that's why those ribosomes are on the membrane, because they translate things that are outside the endoplasmic reticulum, but as the proteins get produced, the amino acid chain ends up on the inside of it. Okay, so once the protein is produced, you know, it folds all up. You know that a protein is just a folded chain of amino acids, and it can travel through the endoplasmic reticulum. So as it's traveling through this network of tunnels, and as it's traveled, various things are happening. We're not going to get into the exact biochemical aspects of it, but eventually the protein is moving through the endoplasmic reticulum, the rough ER, and then it moves through the smooth ER, all the way until it gets to the Golgi bodies. And all sorts of other things happen. I'm oversimplifying a bit, but I just want to give you a sense of what everything in the cell does. So once the protein travels to the Golgi body, it's being prepared for traveling outside the cell, or maybe to the cell's membrane. So what's going to happen is the proteins are actually going to bud out of the Golgi body and carry with it a little piece of the Golgi body, and that's going to be a sphere. So as you can see right here, we have a protein that has budded out, taking with it a piece of the Golgi body, and so it's completely budded out of the Golgi body. Okay, so let's think about what has happened, just to go over it. So mRNA goes to a ribosome that's attached to the rough ER. It is translated into a protein that travels through the endoplasmic reticulum, first through the rough ER, where all the ribosomes are, then through the smooth ER, and the smooth ER has other functions. It also helps to produce hormones and other fatty compounds, but we won't get into detail there. It just travels and connects through the Golgi bodies. Then the Golgi bodies, the proteins can, then through the Golgi bodies, the proteins can bud off and take a little bit of the membrane with it. Now this idea, this structure here is called a vesicle. And let's label that. Okay, vesicle. So you have a vesicle there. And a vesicle is just a really general term for anything small, mostly proteins in the cell that are floating around and are surrounded by their own little mini membranes. The reason why this mini membrane is useful now is because the protein can now float to the outer membrane of the cell, or it can float to other parts of the cell. And then the protein that's surrounded by the membrane can merge with the outer membrane, and it can either get embedded into the membrane or can leave the cell completely, because since the vesicle membrane is made out of the same material as the cellular membrane, they can merge together, and that all works out fine. So the point of the little vesicle membrane is to facilitate the protein getting out of the cell. Okay, so we've already made pretty good progress showing the anatomy of the cell, and there are a few other things that we can throw in there. There are some things called lysosomes, which exist in animal cells, that contain enzymes that can help dissolve other things. So let's draw the lysosome. This is a little larger than it would actually be, but if a lysosome attaches to something else, it is able to spew its enzymes into it, and that usually kills it. So the lysosome contains hydrolytic enzymes. So an example of this in the cells of our body are particularly our immune cells, and our immune cells, specifically of our immune cells, the macrophages. Now a macrophage, which is like a white blood cell, contains many lysosomes, because when the white blood cell wants to kill a pathogen, what it does is it wraps around the pathogen and gets the pathogen inside of it. From there, the lysosomes actually act on the pathogen and release their hydrolytic enzymes, which are able to digest and destroy it. So that's what a lysosome does. Okay? Now in plants you have things called lytic vacuoles. They're really the same thing as a lysosome in terms of their function, that they're really big, they're really big vesicles. In fact, in general, a vacuole is just a big vesicle. So it's just a term for a big membrane-bound organelle. So a lytic vacuole would be a vacuole in a plant cell that stores a bunch of enzymes, and if it were to attach to something else, it would be able to dissolve it. Now there are several organelles that we've talked about within the context of respiration and photosynthesis. Animal cells, everything is called mitochondria, and mitochondria have an inner and outer membrane. So let's draw a mitochondria. Let's get rid of our lysosome and draw a mitochondria. So here we have a mitochondria, and we can see that they have an inner and outer membrane, the mitophage. Okay, so this is where we produce our energy, where sugars are turned into ATP. Now mitochondria actually contain their own DNA, and they can actually reproduce on their own, which makes scientists believe that they once existed as independent prokaryotic organisms. So there's a theory of the origin of eukaryotes that suggests that a prokaryote, like this ancestral mitochondria, might have decided to live in another cell. And that theory is called endosymbiosis. Okay? So the mitochondria are organelles that at one time their ancestors might have been independent prokaryotes. So mitochondria, that's where cellular respiration takes part. And then in plant cells you have chloroplasts. which are a subset of things called plastids. The chloroplasts are the most famous. Maybe I should do that especially in green. Okay, so get rid of this. So I don't have enough space. So here we have a green chloroplast. So we know they have the little thylakoids in there, and that's where photosynthesis takes place. And I do want to make a video on photosynthesis to go into detail on that, but for now let's keep it simple. And just like the mitochondria, chloroplasts have their own DNA and their own ribosomes. So the belief is that they also were once independent prokaryotes that learned to live in symbiosis inside a larger eukaryotic cell. So we're really almost done with the structure of cells, but there are some other things that we can throw in here. Really dealing with plant cells or non-animal cells, we have something called a cell wall, which gives some strength to the outer membrane. And you can kind of think of it in a way that it gives rigidity to the cell. So here we can draw the cell wall, which is going to be past the plasma membrane. And in plant cells, this is really what gives plants their characteristic plant-like features that gives their structure of a plant and gives it rigidity. So the cell wall is actually made of a chemical called cellulose, and you can think of it like a brick wall. So cellulose is going to be these sugar molecules that are assembled in a certain way to be very strong and rigid. And cellulose is actually fiber, so we cannot digest cellulose, so it passes through our body as fiber. Okay, so humans do not possess the enzymes to digest cellulose. Now, humans, well, animal cells do not have cell walls, but we do have these things called microfilaments, or sometimes called actin filaments. And these are basically like little pipes that extend throughout the cell, like little scaffolds, and they basically give structure to the cell. And these actually help, they actually can participate in things like cellular movement and locomotion, and it is known as the cytoskeleton, this network of pipe-like filaments. So just to be complete and make sure we're covering everything, if you watched my video on mitosis or meiosis, you would have heard of these things called centrioles. And to go into detail there, centrioles are right outside the nucleus, and they are right angles of each other to form what is called a centrosome. And you have two of these and two centrosomes, and they basically participate in mitosis, dealing with the synthesis of tubulin, which, as we know, is dealing with attaching to the connect core of the chromosomes and pulling them in mitosis. So, that is about covering everything with cell biology from this simple standpoint. So thanks for watching Stephen Loves Science. Please subscribe, rate, and comment.